Hi everyone, welcome to my channel Capsule. Today we will learn about structure of prokaryotic cells and their functions. Okay, so today's topic is structure of prokaryotic cells and their functions. Okay. So, what is prokaryotic cells? Actually, prokaryotic cells are single-celled, simple organism that lacks of a nucleus or any other membrane-bound organelles. Okay, so it's a unicellular organism that lacks of nucleus or other membrane-bound organelles. Clear to you? Now, we need to understand prokaryotic cells and eukaryotic cells share some common components, mainly 4 common components. Okay, which kind of common components? Like first one is plasma membrane or also known as cell membrane. We can see in prokaryotic cells and also in eukaryotic cells. Okay. Second one is cytoplasm. Okay. And the third one is DNA. And the last one is ribosomes. Okay. So these are the 4 components which are common in prokaryotic cells and also in eukaryotic cells. And others which are only found in prokaryotic cells like that are cell wall, Pili, Fimbriae, Flagella, etc. Okay. You need to be clear about all the components. Everything you need to know for your exam point of view, for your knowledge purpose, for any kind of entrance or competitive exams. Okay. Let's start it step by step. So first one, we will learn about capsule. What is capsule? Actually, it is the most outer protective covering which is sticky in nature, and it is actually used to fix the bacteria into the outer environment and also known as slime layer. Okay, so it is the most outer protective covering okay, sticky in nature and also known as slime layer, which is mainly composed of polysaccharides, polypeptides, or it can be both. Okay. Now, we need to be clear about Glycocalyx or capsule or slime layer. We have learned about the term Glycocalyx. There is a little bit difference among them. Now, what is Glycocalyx? Actually, Glycocalyx is the thin, high molecular weight secretory substances or granules that are present in the external part of cell wall of bacteria. Okay. So glycocalyx actually, if this is the bacterial cell, okay, and this is the cell wall, glycocalyx are the secretory substances which are present in the upper part of the cell wall like this. Okay, and if this glycocalyx is organized in a tight matrix and rigid in nature, that is known as capsule. So what is capsule? When the glycocalyx is rigid, that is capsule. Now, if the glycocalyx is more easily deformed and loosely attached to the cell wall, that is known as slime layer. Okay. So slime layer is when the glycocalyx is loosely attached. Okay, that is glycocalyx. Sorry, slime layer. Okay, and this capsule is mainly responsible for the virulence factor of bacterial cells. So responsible for virulence factor. That is capsule. Now we need to be clear about cell wall. Second one is cell wall. It is also an important topic or part of the bacterial cells. Okay. Actually, this is the next layer of capsule. If this is the capsule, this is the cell wall part. Okay. And major component is peptidoglycan or murein. Okay. And only found in the bacteria. So, major component is murein or also known as peptidoglycan. Okay, and structurally this peptidoglycan layer consists of meshwork of long chain alternating molecules of NAM and NAG. So this is a meshwork of alternating structure of NAM and NAG. What is the full form of NAM? NAM means N-acetyl muramic acid and NAG means N-acetyl glucosamine. Okay. There is a little bit difference in gram-positive and gram-negative bacterial peptidoglycan structure. Okay. We will look it. Okay. So, let's draw something. Okay, this is the peptidoglycan chain. Okay, let's consider this is gram positive and this is gram negative. Okay, now we will look the difference. We can say like this is NAM, okay, and this is the NAG. Okay, and a tetrapeptide is formed from this, okay, NAM part. That will connect another chain. It means two peptide chain are connected with this tetrapeptide. Tetra, but I am drawing here a long chain. Actually, these are tetrapeptide. 4 peptide is linked together. Okay. Now is the difference. In the gram-positive bacteria, these tetrapeptide connected through a pentapeptide chain like this. Okay. But in gram-negative bacteria, actually they directly attached with this tetrapeptide with each other. Okay. That is the difference. That is tetrapeptide. That is pentapeptide. It means in gram-negative bacteria, pentapeptide is absent. In gram-positive bacteria, pentapeptide is present. That is the difference of gram-positive and gram-negative bacteria. One thing you need to know, carbohydrate, one teichoic acid type of structure which is present, this type of structure which is present in the gram-positive cell wall. Now, what is teichoic acid? Actually, it's a carbohydrate chain that extended through or beyond the peptidoglycan layer. Okay, through the layer or beyond the peptidoglycan layer. Okay, so this is the teichoic acid chain, and this capsule actually provide the protection. Okay. Now, the third one is cell membrane, also known as plasma membrane or cytoplasmic membrane. Okay, actually it encloses the cytoplasm and internal structures. Okay, so it encloses cytoplasm and internal structures. Okay, it actually exhibits selective permeability. It means which allows some molecules to enter or leave the cell and restricting the passage of others. That is selective permeability. Okay, what I have said, selective permeability means which allows some molecules to enter or leave the cells but restricting the passage of others. That is selective permeability. Okay, so it exhibits selective permeability. That is important topic. Okay, now glycoprotein and glycolipids are the composition, and their chemical composition actually varies among archaea bacteria. So what is the chemical composition? That is glycoprotein and glycolipids. Among the membrane transport mechanism, bacterial cells actually shows simple diffusion process, facilitated diffusion, active transport that are common. And I am not describing in details because already I have made a short video on this. Okay, so what are the membrane transport mechanism? That are like simple diffusion, facilitated diffusion, and active transport. Okay, okay. Now the next structure is the nucleoid. Okay. So what is nucleoid? Actually, it's a prokaryotic chromosomes that are typically circular, haploid or unipaired and not bound by complex nuclear membrane. So these are circular, haploid and not bound to complex nuclear membrane. Clear? So the DNA and DNA associated proteins are concentrated within the nucleoid region of the cell. Okay. Prokaryotic cells may also contain extra chromosomal DNA or the DNA that is not the part of chromosome, and they are small circular and double stranded DNA molecules. So extra chromosomal small circular and double stranded DNA that is known as plasmid. Okay, and it carries genes that are responsible for antibiotic resistance and takes part in important survival mechanism. Clear? Now the fifth one is ribosomes. Okay, so these are the structures that are responsible for protein synthesis in the prokaryotic cells, and prokaryotic ribosomes are called as 70S ribosome. Okay, responsible for protein synthesis, and these prokaryotic ribosomes are also called as 70S ribosomes. Okay, what is S? S means the Vedvark unit. Okay, Vedvark unit is a measure of sedimentation of an ultracentrifuge which is based on size, shape, surface qualities of the structure being analyzed. Okay, now the next is inclusions. Okay, that is inclusions. Some prokaryotic cells have the ability to store excess nutrients within the cytoplasmic structure that are known as inclusions. So what are inclusions? When they stores excess nutrients. Okay, and they store nutrients like in a polymerized form. Okay, so they store in polymerized form. Why they store in polymerized form? Why? Because it reduces the buildup of osmotic pressure that occurs as a cell accumulated solutes. Okay, now it can stores polymerized form. What it can stores? Like glycogen, starch, voluten granules or metachromatic granules like this. Okay, so they can stores glycogen, it can be starches or it can be voluten granules, etc. Now the next topic is the seventh one is endospore. That is very much important. Okay, bacterial cells are generally observed as vegetative cells. Okay, but sometimes some bacteria can form endospores also. What is endospore? It is a structure that essentially protects the bacterial genome in a dormant state when environmental conditions are unfavorable. Okay, so it's a structure that protects the bacterial genome from the unfavorable environmental conditions. Unfavorable environmental conditions. Okay? And the process by which these vegetative cells transform into endospores is called as sporulation. Okay, so the process is process of endospore formation. Sporulation. Clear? And it generally begins when the nutrients become depleted or environmental conditions become unfavorable. In the growth curve, it can be seen in the stationary phase. Why bacterial pathogens are difficult to destroy? Because their endospore are so hard to kill. So, some sterilization methods are applied. Now, the next is eighth part is Fimbriae and Pili. Actually, bacterial cells have protein appendages that embedded within cell envelopes that extend outward and allowing interaction with the environment. These appendages can attach to other surfaces, transform DNA, or can provide movement. These includes Pili and Fimbriae. So, what is Pili and Fimbriae? These are appendages that embedded within cell and extend outward, which allows interaction with the environment. Okay, Fimbriae actually refers to the short bristle-like proteins projecting from the cell surface that enable a cell to attach to the surface of another cells. Okay, so Fimbriae actually short bristle-like proteins that enable cells to attach to the surface of other cells. Okay, and Pili in singular, that is pilus, refers to the longer, less numerous protein appendages that helps the cell in attachment during the transfer of DNA between two cells. That's why they are known as Pili or sex pili. Pili is, P-I-L-I. Pili is longer, less numerous protein appendages that helps the cell cells in transferring DNA. Okay, that's why they are known as F. pili or sex pili. And the protein that is present is known as pilin protein. Clear? Another part is the last part, that is flagella or flagellum. Actually, these are the structures that used by bacterial cells for their movement in aqueous environment, and they act like propellers. So, propeller-like structure, that is responsible for movement of bacterial cells. Clear? They are stiff, spiral filament and composed of flagellin protein that causes spin in solution. Okay? So they composed of flagellin protein which produces spin in solution. Okay, so this is the end of my lecture. If you like my video, please do like, comment, share and subscribe. Thank you for your patience.